Good morning to all the students. So yes, today we are going to learn about those organisms or those species which are not included under five kingdom system of classification. In the previous class, we have dealt clearly on all the kingdom systems of classification, explored all the systems from 2 kingdom to 6 kingdom system of classification, and then we have learnt the systems of classification from plant kingdom as well, that is artificial system, natural system and phylogenetic system of classification. Today we are going to explore those domains which are not included under five kingdom system of classification, which includes viruses, viroids, prions and lichens. These are the organisms which are not included in any of the kingdom under five kingdom system of classification. Firstly, talking about viruses. Remember, this is a major objective which is asked quite a few times in your NEET, like from any of this entity you will get a question in the alternate years of the NEET exam. In every alternate year of the NEET paper, you can see the question from this concept of biological classification, either from viruses, viroids, prions and lichens, which are not included in five kingdom system of classification, and in this class we are going to learn completely about all this. First, about viruses. Viruses are infectious agents. They are infectious agents, non-cellular organisms. And do you know, virus is also an exception to cell theory. Virus is also an exception to cell theory, because cell theory is applicable to those cells which has a cell membrane and a nucleus and a cytoplasm. It has a cell membrane, nucleus and a cytoplasm. Only to such cells cell theory is applicable. But when you see the components of viruses, viruses just has a protein coat, and genetic material inside it. It just has a protein coat and a genetic material. So, viruses are infectious agents. These are non-cellular organisms. They are the exceptions of cell theory, and they are the connecting links between living and non-living entities. Okay. Viruses are the connecting links between living and non-living entities, and they are termed as what? Infectious agents or simply living entities. Viruses outside the living cell, they can sustain, but they cannot replicate. They cannot replicate or increase its number Inside the host or living cell, virus replicates and increases its number. Inside the living cell or host cell, virus undergoes replication. Clear, right? So, here viruses are not included under five kingdom system of classification. How can you describe virus as infectious agents, non-cellular organisms, and viruses are also the exceptions of cell theory. They are connecting link between living and non-living entities. Inside the host or living cell, they can replicate and increase their number. Outside the living cell, they can just sustain, but they cannot increase their number. Okay. So, this is all about viruses. And now, who discovered virus? In what form virus is first identified as? It is D.J. Ivanovsky, who first discovered virus, that is tobacco mosaic virus. He didn't named it as tobacco mosaic virus, it is named by Bajering. This is named by Bajering, but this effect of disease is identified by D.J. Ivanovsky, tobacco mosaic virus, wherein from the affected plant, he has extracted the fluid, he has extracted the leaves of the affected plant, that is tobacco mosaic plant, and he has made a fluid out of it. After centrifugation, he has passed that through bacterial filter paper. He has passed it through bacterial filter paper and observed that these molecules have passed on through bacterial filter paper. And next it is W.M. Bajering, who has transferred this fluid, which has passed out from bacterial filter paper, and he has subjected this to a healthy plant. He has subjected this fluid to a healthy plant. Fine, right? He has subjected this fluid to a healthy plant and observed that the healthy plant also got the disease. Got it, ma? First it is DJ Ivanovsky who has identified tobacco mosaic disease caused by a virus, that is tobacco mosaic virus. He didn't named it as virus, but he has identified one thing, that the fluid which is passed from the infectious leaves has passed through the bacterial filter paper. Later it is M.W. Bajering observed that or transmitted the fluid which is passed out from the bacterial filter paper. He has transmitted it to a healthy plant and observed that the healthy plant is also subjected to the disease, and this he has named it under the concept of contagium vivum fluidum. Contagium vivum fluidum. Is it clear now? Is the voice clear now? Yeah. Next it is W.M. Stanley who has crystallized this virus. It is W.M. Stanley who has crystallized the virus. Crystallized the virus particle. We know this concept that virus has a protein coat and a genetic material, right? This protein coat is subjected to crystallization, and this concept is first introduced by W.M. Stanley. So, three discoveries you have to remember, that who is the first person who identified tobacco mosaic disease? The first person who identified tobacco mosaic disease is DJ Ivanovsky. He has passed it through bacterial filter paper, and next it is M.W. Bajering, it is W.M. Bajering who has transmitted this fluid to a healthy plant and given the concept of contagium vivum fluidum, and then it is W.M. Stanley who has crystallized the virus particle. This is about discoveries that we have to learn. And then virus consists of what components? What are the components of viruses? Virus consists of three main components. First is envelope, second protein coat, and third component is genetic material. Genetic material can be either DNA or RNA based on the mode of infection that virus is causing or based on the host cell that virus is invading. It can be either DNA or RNA. Got it? So, virus consists of what components? Envelope, protein coat and genetic material. Enveloped viruses are optional because when we have to consider the concept of envelope, we have to classify the viruses into enveloped viruses and non-enveloped viruses. So, envelope will be having for only few viruses, not all, but protein coat and genetic material is mandatory for all the viruses. Got it, ma? So, first enveloped viruses, here if I can give you one example of herpes virus, the envelope is having one protein. This is envelope protein which is pinpointed like this, which is covered by an envelope. Below the envelope there is a protein coat and inside there is genetic material. Best examples that I can give you for enveloped viruses includes HIV virus and herpes virus. These are the enveloped viruses. Next is what, ma? Capsid. Capsid is the protein coat. Capsid is nothing but protein coat of the virus. Protein coat definitely has amino acid chains, right? What is a protein? The chain of amino acids. So, capsid is what? Protein coat. The subunits of this protein coat are called as capsomeres. Okay. Like how we will say amino acids are building blocks of protein. Here those amino acids are addressed as what? Capsids, because these are the units of capsid. Capsid is what? Protein coat, and the units of this capsid is what? Capsomeres. These capsomeres, they are arranged helical, or they are arranged in polyhedral fashion, or they are arranged in the geometric forms. In any ways these capsomeres can be arranged. Helical form of arrangement you will see it in tobacco mosaic virus. Okay, helical form of capsomere arrangement you will observe it in TMV. So, in this slide what did we learn? Virus components, which include envelope, protein coat and genetic material. Envelope will be having HIV virus or herpes virus. Capsid consists of capsomeres. They are arranged in any of the forms like helical, polyhedral or geometric forms. Got it? This is about the protein coat. Next is nucleoid. Nucleoid can either be in the form of RNA or DNA. Nucleoid can either be in the form of RNA or DNA. Animal infecting viruses will have what type of genetic material? Animal infecting viruses will have double stranded DNA as the genetic material. For example, pox virus and herpes virus. Plant infecting viruses will have single stranded RNA as the genetic material. Plant infecting viruses has single stranded RNA as genetic material, which includes the examples like TMV and HIV. Next, bacterial viruses or bacteriophages will also have double stranded DNA, but most of them will be circular double stranded DNA, not in the linear fashion. They will be circular double stranded DNA. So, nucleoid can either be RNA or DNA, but not both. Double stranded DNA you will observe it in animal infecting viruses like pox virus and herpes virus. Plant infecting viruses will have single stranded RNA. Examples include tobacco mosaic virus, not this. This is an exception. This is not a plant infecting virus, but this is an exception which has single stranded RNA. Bacterial viruses and bacteriophages will also have double stranded DNA, which is in the circular format. Next, viral infections. If you see what type of viral infections you will observe in animals and plants. Viral infection in animals include mumps, chickenpox, herpes, influenza and AIDS. These are the infections, viral infections that are observed in animals. In plants, tobacco mosaic disease is caused by tobacco mosaic virus. Cauliflower mosaic disease is caused by cauliflower mosaic virus. Okay, this is the basic viral infections that we have to see, and the symptoms that which are marked in the NCERT, very important. These symptoms you have to fix it in your mind. Okay, few variations will be there for bacterial infections and viral infections. Here we go with the symptoms of viral infections. Symptoms of viral infections include, first is what? Mosaic formation, leaf rolling. Leaf will be rolled towards inner side. Here you can see leaf rolling. Leaf rolls towards the inner side. Leaf curling and yellowing. Here you can see leaf curling and yellowing. Vein clearing, dwarfing, stunted growth. Vein clearing, dwarfing and stunted growth. These are the symptoms of viral infections. What are they? Mosaic formation, leaf rolling, leaf curling or yellowing, vein clearing, dwarfing and stunted growth. These are the examples of symptoms of viral infections. Next is tobacco mosaic virus. Tobacco mosaic virus is first observed by D.J. Ivanovsky. We have discussed just now, right, in the discoveries part, that tobacco mosaic virus is first observed by whom? D.J. Ivanovsky. And as it is plant infecting virus, what will be the genetic material? Genetic material is single stranded RNA. Genetic material is single stranded RNA. And these are the protein subunits which are known as capsomeres. Capsomeres. These capsomeres are helically arranged. Capsomeres are helically arranged. These capsomeres are helically arranged and they build up to what? Capsid, which is the protein coat. This is an elongated rod-like structure. TMV is an elongated rod-like structure with 3000 angstroms long and 180 angstroms in diameter. This is 3000 angstroms long, 3000 angstroms long and 180 angstroms in diameter. Got it? Now, the molecular weight of the TMV will be 39.4 into 10 to the power of 9 Daltons. And how these capsomeres are arranged? Helically. Single stranded RNA has how many? 6400 nucleotides, and this complete capsid is made up of 2130 capsomeres. Got it? RNA has how many nucleotides? 6400, and capsid will have how many capsomeres? 2130, which is an approximate number. The ratio between nucleotides and capsomeres will be what? 3:1. If they are giving you the ratio between capsomeres and nucleotides, it will be 1:3. Got it, right? This is all about tobacco mosaic virus. And now if we have to learn about bacteriophage, bacteriophage is commonly noted as tadpole virus. Bacteriophage is commonly addressed as what structure? Tadpole structure, which is having double stranded DNA in the head center. Okay? The main components of it includes, first is head, followed by collar, sheath and tail fibers. Head, collar, a sheath and tail fibers. Got it? So, the double stranded DNA is present where? Inside the head, and this is of what structure? Tadpole structure. It has a polyhedral head and a helical tail. Okay? It has a polyhedral head and a helical tail. Head has double stranded DNA, it has neck, tail region, helically arranged protein enzymes are present. Tail region has helically arranged proteins. Tail fibers at the end, it will have what? Lysozyme. Tail fibers at the end consists of lysozyme. Got it? So, this is about bacteriophage, which is a tadpole structure. It has polyhedral head and a helical tail. The main components of this include head, collar, sheath, tail fibers, tail and tail fibers. At the end of tail fibers, the enzyme which is present is lysozyme. This lysozyme will be helpful in cleaving the bacterial cell wall and injecting the DNA directly into the cytoplasm of bacterial cell. Clear, right? This is about bacteriophage. Now we will learn about subviral agents. The prerequisites of virus, it should have both genetic material and a protein code for sure. If at all the particles which just has the genetic material, for example RNA, but protein code is absent, such particles are addressed as viroids. Few infectious particles which just has protein code, which is abnormally folded, but it does not have the genetic material, such particles are known as prions. Got it? So, what are viroids? The genetic material without the protein code. For example, RNA without the protein code is known as viroid. Prion which just has the protein code which is abnormally folded, but it does not has the genetic material, those are known as prions. Now, we learn both about viroids and prions in detail. This is a microscopic view of a viroid. Viruses without the protein code are known as viroids. Who described about this first? This is first described by T.O. Dynier. This is first described or discovered by T.O. Dynier in the year 1971. From what disease? Potato spindle tuber disease, where it just has single stranded RNA. That single stranded RNA is tightly folded. Single stranded RNA is tightly folded into circular or linear structures. Okay? Single stranded RNA is tightly folded into circular or linear structures, and this has low molecular weight. This has low molecular weight. So, viroids, these are the viruses without the protein code. It is first discovered by T.O. Dynier in the year 1971, and the cause is potato spindle tuber disease. It has single stranded RNA which is tightly folded to form circular or linear structures, and it has low molecular weight. Now, about prions. I already told you prions are what? Abnormally folded proteins. Here we see abnormally folded proteins. Nucleic acid is completely absent. There is no genetic material at all. The cause is neurological disease. Prions will first affect what? Neurons or brain. So, it is what? Neurological disease. For example, this causes scrape disease in sheep and mad cow disease in cattle. This is also addressed as bovine spongiform encephalopathy. Bovine spongiform encephalopathy. Bovine spongiform encephalopathy, which is mad cow disease. This gives an analogous variant, that is CR-Jacob disease, that is CJD in humans. Got it right? So, prions are what? Abnormally folded proteins where the nucleic acid is completely absent. This causes neurological disease. For example, scrape disease in sheep. Same analog will cause mad cow disease in cattle, which is addressed as bovine spongiform encephalopathy. Analogous variant to this for humans include CR-Jacob disease, that is CJD. Now, if you consider the molecular weight of genetic material will be higher, right? So, molecular weight of virus is higher, which consists of genetic material plus protein coat. Compared to virus, viroids has less molecular weight because this just has genetic material. Genetic material is made up of nucleotides and this is RNA, right? Single stranded RNA. Each nucleotide further has components sugar, phosphate group and nitrogen base. So, this has more molecular weight when compared to prions. Prions, as they are abnormally folded proteins, they are just combined with amino acids, which has less molecular weight. Prions has less molecular weight than viroids and virus. Remember the hierarchy, which is very important, quite a times asked about molecular weight in our exam. Now about lichens. We all know from very younger standards that lichen is a symbiotic association between algae and fungi. But what is the percentage of algae? What is the percentage of fungi? What is their association? We will learn it now in detail. So, lichen is symbiotic association between algae and fungi, in which the algal component is just 5%, which is known as phycobiont. Got it? Fungal component is in bulk. 90 to 95% of the lichen component is fungi, which is mycobiont. Got it right? So, algal component is phycobiont, fungal component is mycobiont. Algae will prepare nutrition to the fungi, it will prepare the complete food and nutrition to the fungi, and fungi will provide shelter, it will provide shelter to algae. Got it? Algae provides food or nourishment or nutrition to fungi, fungi provides shelter to algae. And these lichens are sensitive to pollution. This will be the association. See, can you see this is the complete association of fungi in which you can see very less percentage of algae. They are very sensitive to pollution, which are addressed as very good pollution indicators. Okay? They are very good pollution indicators. Most of them are sensitive for air pollution, which is mostly sulfate pollution sensitive. They can easily detect the pollution of sulfate and nitrates, okay, which is very sensitive to air pollution. They do not grow in polluted areas. To dig a bit, how can they identify the pollution? How are they unable to grow in polluted rich areas? I will tell you the reason. It has 5% of algal component, right? 5% of algal component, where the algae has chlorophyll inside it. It has chlorophyll inside it. Chlorophyll consists of a porphyrin ring, and a phytol chain. Porphyrin ring and a phytol chain. This porphyrin ring at the center, it is incubated with magnesium. Got it? at the center magnesium is connected to the porphyrin ring, to all carbons of porphyrin ring. So, here when pollutants, when the lichen is subjected to air pollution or pollutants, magnesium will be interacting with either nitrates or sulfates, and this will be converted to magnesium nitrates or magnesium sulfates, magnesium nitrates or magnesium sulfates, which will cleave the bond here, and this will disintegrate the structure of the chlorophyll. This will disintegrate the structure of chlorophyll. This will disintegrate the structure of chlorophyll. Thereby, the complete process will be inhibited. When algae stops providing nutrition to the fungi, then the complete lichen system will be collapsed. Got it? So, though the algal percent is 5%, if it is collapsed, if it is structurally disintegrated, adjoining and forming bonds with magnesium nitrate or magnesium sulfate, when the porphyrin ring got disrupted, algal structure got disintegrated, wherein this will completely disintegrate or collapse the structure of lichen. Got it? So, that is how lichens are sensitive to pollution. This concept will not be there for your NEET or NCERT, but learning this will be interesting and you will get to know how lichens are good pollution indicators. So, this is the story behind it. And the key takeaways of today's session includes, from biological classification, we have learnt about virus, viroids, prions and lichens. About all the 4 components we have learnt in detail. Got it right? So, that is all for today's session. Yeah, that is all for today's session, and in the next class we will analyze few more concepts from core botany chapters. Got it? So, that is all with today's session, and thank you.